You are listening to the Mary Jane Society podcast, where you will meet entrepreneurs, cultivators, scientists, doctors, and inventors in the cannabis industry.
I'm your host, Pam Schmiel, marketer and publicist in the cannabis industry. Today we meet Dr. Dedi Meiri. He heads up the Laboratory of Cancer and Cannabinoid Research at Technion, Israel's highly regarded Institute of Technology. He comes from a highly diverse scientific background in biochemistry, plant biotechnology, and cancer research, and has about 50 research scientists working under him. We discuss the complexities of the endocannabinoid system and Dr. Meiri's painstaking work to document numerous combinations of cannabinoids to determine which ones unlock specific receptors to treat diseases like cancer. Does the endocannabinoid system hold the answers to understanding how cancer works in the body and how to kill it? We talk about this and more. Let's meet Dr. Mieri. I'm very excited to learn about precision medicine and where we are with that in as far as our endocannabinoid system. And just if you could just tell us what research you are working on in your lab, just to give us a quick overview.
Okay. So my lab is expert in a kind of a drug discovery, mainly to cancer, but for other indication. We are looking on plants that are in herbal medicine are quite known to be effective on humans, but still the molecules or the active ingredients are not clear and the mechanism of action is not clear. Cannabis is a good example for that, thousands of people use it as a medicine, but actually, it's clear to us today that we're looking on two major cannabinoids, THC and CBD, when there are more than 160 others, and the activity is sometimes more complex. It's a combination of molecules, what we call the entourage effect sometimes, and sometimes these are the rare cannabinoids that are doing the effect, even though they're not expressed in high levels, or we just don't see them because we don't know to look on them. So sometimes you have what we call the rare cannabinoids or minor cannabinoids, but actually it's not minor. It can be in few percentage in the extract, but people, because they don't know to analyze that, they don't see it, they don't know it's there. So we have few examples that we define activity on these diseases in cannabis that we found cannabinoids which are unknown, but very active, and they are the reason for the activity. We're doing that not just with cannabis, we're doing that with other known plants from, psychedelic mushrooms or other functional mushrooms or other plants that in herbal medicine are very known to be effective. But when you look on the data, people all look on the major molecule. In psychedelic mushrooms, psilocybin mushroom, we're looking at psilocybin when we can see more than 60 different terpenes, which are similar to serotonin and doing effect. So it's similar. Then we focus on cannabis. Cannabis is a unique plant because it's active on the endocannabinoid system in our body, that actually every cell in our body harboring the endocannabinoids have receptors for the endocannabinoids and can produce the molecule that's similar to the phytocannabinoids in our body, which are the endocannabinoids. So the endocannabinoid system, it's very complex system in our body. We can analyze from blood samples more than 100 different endocannabinoids. People usually know anandamide and 2-AG, but there are more than 100. And there are many receptors. We are usually naming CB1, CB2, but my lab look on more than 35 different receptors. And we have in our body more than 220 different types of cells, neurons, skin cell, heart cell, breast cells, whatever. Each one of them will have a different combination of receptors. So you will know to communicate with different endocannabinoids. Sometimes it's a combination of endocannabinoids, sometimes it's one endocannabinoid, but in every cell in our body, the action is very localized and very specific. I will give one example. If we're talking about sleep, endocannabinoids are very important in sleep. But sleep, there is actually 4 major actions: is how fast we are falling asleep, getting tired and falling asleep, how deep is your sleep or how strong is your sleep, whether you are waking up from every noise or something like that. And think about evolutionary, it's important if you are sleeping outside and there are tigers around you, you need to know to wake up from the right noise and not, okay? And whether your sleep is effective sleep, whether you're going to REM sleep or dream sleep, because in this moment when your sleep is deep, your body produces different hormones like building bones and other things, and waking up. And actually, it's been activated in 4 different areas in our brain. And in each area, different endocannabinoids are doing a regulation of this. So when you turning off the light and quiet and in your bed and getting to sleep, different endocannabinoids will be released, preparing you to get to sleep. So even in our brain, in different areas in our brain, these are different endocannabinoids. So taking just blood sample, it's not really showing what's happened now in your left areas or your amygdala in the brain. So it's very complicated. And cannabis is just mimicking them. So which cannabis, which combination of cannabinoids mimicking the combination in the amygdala, which one are mimicking what's happened in your heart or in your immune system? This is what we are learning, testing, and trying to understand, trying to break this complexity and understand it. And I will say 20 years ago, we couldn't do that without computers, without our analytical abilities today. So if you're taking blood samples today and run it on the equipment that we have, you can see more than 1,000 different compounds or something like that without computers, without very strong analytical tools. 20 years ago, you couldn't look on 1,000 different molecules in 1 hour. You know, it's you're running it, separating it, see the structure, send it to the cloud, coming back, telling you these are the compounds. This is something that would take us 40 years, which now takes 1 hour. So the technical ability opened amazing opportunities to understand this complexity of the nature of the plants that affecting us, which we just couldn't do years ago.
And are you also, is your team also developing the technology to be able to analyze this?
Yeah, so that's our claim of fame. So we were the first one that developed the methodology and the equipment to analyze all the active compounds in cannabis. So when I started to work, people could analyze the 11 different phytocannabinoids, and we create a library of 165 to be able to analyze 165 phytocannabinoids from the plant. And then we did it for the endocannabinoids in our body. Before that, we couldn't.
And then matched
now
it.
We can start to match it.
I see. Okay. Wow, that's amazing. So then how are you? Okay, so your claim to fame is developing this technology. And then how are you after you identify them, like you said, once it goes to the brain, you know, and it starts working, I guess, in the nervous system and things like that. So how are you then going deeper and understanding like what you were just talking about?
So first of all, I'm a cell biologist. I'm coming from background from plants and from cancer, and I'm not a chemist. So I had to do that in order to actually to ask the questions, but because there was no knowledge, but I wasn't an expert for analytical chemistry and that. I had to develop that in my lab. And I was lucky to have a very good people that come to work for me to be able to teach the team and to do that. And how are we doing that? Usually we're starting from a basic knowledge. We know that these cannabis strain affecting sleep, okay, from database that we acquired from patients and other things. So we will take that strain that we already know is affecting sleep, and now we will develop methods to start to find the molecules that are doing that. Whether we will grow cells on a plate or we will use a mice or whatever. So we have, let's say, a mice model that we see that we give them the cannabis and affecting sleep. And then we started to work on the cannabis to start to dissect the cannabis. Let's say there is 500 active molecules. We will chop it to 4 pieces, to 125 in or 150 in each, piece. Come back to the mice, see which combination is working, and then we narrow it down until we found the minimum compound that doing the effect. Whether this is 1 compound, 3 or 7, we'll find the combination that doing the effect. After we find the combination, we're starting to ask how, which receptor they're binding, what's happened in the cell, which other active compounds being released, and then this is years of work. Okay, so I would say from the moment we have a plant, okay, a strain or an extract that we know that working, to understand the mechanism of action, which molecules are doing the effect, and really what's happened, it's around 5 to 6 years. It's not, it's okay.
By the time you back and forth and process of elimination and all that.
Yeah, so to find the molecules will take few months, but then to understand which receptor they're binding, what is the cell biology, what is the, which pathways in the cells being activated, and exactly what's happened, this takes years. But this opened, what we started to say, this opened the ability to do a personal medicine. Because if you know which receptors is binding, what need to be in the person, which receptors need to be expressed, or in which areas of the body it will be act, or something like that, you can start and look deeper. I will give one example, okay? We worked on breast cancer. In breast cancer, there are types of breast cancer. The major one called estrogen plus. The cells, the cancer cells need hormone called estrogen in order to grow. So one of the ways to fight it is to reduce the estrogen hormone or to block the estrogen receptors that reading the hormone. So there are drugs today that blocking the estrogen receptor, like amoxicillin. But we saw that cannabis is affecting this cancer, and when we started to learn, we defined specific molecule that reducing the expression of their receptors. But when we learned how it's doing that, we understood in which other diseases this molecule can be affected, whether this is endometriosis or other diseases that related to estrogen and not just the breast cancer. But before understanding which molecules and exactly how it's activated, we couldn't guess in which other diseases it will affect. So sometimes you're finding something in skin cell, but you say, just a moment, if it's working like that, in Alzheimer's, it would be very important. Sometimes for, I will say, a normal person, it looks very far, but for a scientist that understand the mechanism of a specific disease, and then you can match it, you can say, just a moment, if it's blocking the immune system to move like that, in this disease, it will be very important. Then you're going there and discovering things that are unknown and more personalized.
I want to ask you more about the cancer, but I just want to ask you first, how does, or like if you made a precision medicine for, say, cancer, I mean, maybe that's too broad, or for sleep, like what you were explaining earlier, like once it goes to your brain and the different types of sleep, like do you think you could create a medicine that would it work on everyone, or is everyone's DNA affecting the interaction of the endocannabinoid system too much to be able to make precision medicine?
So DNA, there is no medicine that's working on everybody by definition, because unless if you're killing something, it's dead. But unless that, chemotherapy, it's throwing a bomb that kill everything that divided. So it's but we learned that it's also not working on everybody. We are different. Every person is different. It's not just the DNA. The DNA is coded how to produce proteins, but you have the same DNA in your skin cell, in your heart cell, in your neurons. It's the same DNA, but they look very different because your body is going to the DNA and reading how to produce specific protein in the skin cell, but it will read the different thing. Usually when I'm teaching on DNA, I'm teaching in medical school and in the biology school, and I'm teaching the basic on DNA. I'm saying that DNA, it's a cooking book how to produce protein, but it's a big book with chapters. We call them chromosomes or whatever. Okay, and in them there are recipe. Each recipe we call it the gene, and a gene it's a recipe how to produce a protein. So your skin cell will go and read chapter 13 and produce specific protein that you need in order to be a skin cell. And your neuron cell will go to chapter number 2 and chapter number 5 and read there. So even you have a cooking book, you can make a meat from chapter 13 or so it's very important in which gene your body is read and how and things like that. It's not just the DNA. This is first. But when we're talking about personal medicines, first of all, important what disease, what is the phenotype, what is the structure of the disease. So if we say, let's say breast cancer, there are different types of breast cancer. So if you have an estrogen plus breast cancer, you need to block the estrogen. But if you have, a triple negative, it wouldn't help. So first of all, we need to understand the disease, but it's not just cancer. I can talk about other diseases like epilepsy, okay? There are different types of epilepsy. We need to know what is the type, what is the mechanism of the disease, and then to know how to block them and to affect them. On the other side, my lab have more than 1,000 different types of cannabis. Which one of the cannabis you're matching to the so there are two sides of the coins here. It's to match the right plant, I will say that in our case, okay, to the right phenotype or the right type of disease. And I will give another example from cancer. We're treating today cancer. One of the major treatments for cancer, or the most innovative, is actually to activate the immune system to attack the cancer. It's called immunotherapy, okay? The cancer cells are normal cells that become crazy. In every moment, we have more than 2,000 cells in our bodies that it's happened to them. In my body and in your body, every moment. All these cells will kill themselves. They're testing themselves. They see something goes wrong, they will kill themselves. They call it apoptosis, the cells committing suicide. And if they are not committing suicide, our immune system will see that something is going wrong and will kill them. Cancer cells know how to avoid that. It's called immune suppressive. They suppress the activity of the immune cells. And they are doing two things. First of all, they're fooling the immune cells. They are presenting a protein. They're putting a note saying, hey, don't attack me. I'm okay. I'm a normal cell. Don't attack me. Okay? This is one thing they're doing. Second thing, they release specific molecules in their surrounding. We call it the microenvironment of the cancer. They're changing the profile of the immune cells that will come. They kind of breaking the fuel for the immune cells to attack them. So they're weakening them. So the drugs today, the major drugs, which we call immunotherapy, are blocking the ability to say, I'm normal, don't attack me, okay? But these cells still will release molecule that will suppress the immune cells, okay? So this is one side. You have a patient that we see in this patient specific proteins like PD-1 that they are presenting, that you can treat with this medicine. But now the question, how the cannabis interact with it? We know from other indication that cannabis is downregulate or suppress the immune cells when they are in hyperactivation. If you're a normal person, and taking cannabis, it won't affect your immune system. But if your immune system is in hyperactivation, let's say in inflammation or other things, the cannabis is reducing this activity. This is the reason cannabis is very good for autoimmune disease, for a inflammatory bowel disease like Crohn, colitis, and other. And patient taking for Crohn or for autoimmune cannabis, because it's downregulate the immune system to attack. But now, if the medicine that we are giving, immunotherapy, is activating the immune system, do you want to reduce it? This is the question, right?
Right.
So we test that. We did 4 clinical trials, and in 2 of them we saw that the cannabis didn't really affect the activity. In 2 of them we saw that the cannabis really reduced the effectivity of the medicine, which is very bad. Okay, we don't want to reduce. So now we ask, okay, which strain, which cannabis the patient take that hurt the treatment, and which cannabis they take that didn't affect or even improve. And we started to study that. It was 5 years work. And in the end, we defined specific strain with specific molecules that actually working in a synergism and helping the immunotherapy and not hurting. So what we defined that there are molecules in the cannabis, we said that the cancer cell doing 2 things, presenting a protein and releasing molecules that reducing the activity of the immune cells. On this arm, on presenting a protein, we're giving a medicine, Keytruda or other medicine. The cannabis, the right cannabis, if you give the right cannabis, will affect the cancer not to release the other molecules. So you actually, if you give the right cannabis, you are blocking the ability of the cancer to defend totally from the immune system, and then it will be more effective. But we must know which cannabis to give the patient. You can give a cannabis that will hurt the patient. You can give cannabis that will help the patient. And this is already almost personal medicine for me, because you need to know what the person, what is the disease, what exactly you're giving him, and now which cannabis to give him. And I think we are going in that direction. We're starting to learn and to understand, to fine-tune the treatment to the patient. And this is a new age in treatment, and I think and hope in herbal medicine in total. Okay, so we know that there are other herbal medicine that if you want to we know that there is a plant, I hope it's the same name in English, Archinicea. You know Archinicea? Maybe I'm not saying that in
Say it again.
Archinicea. Maybe I will give a different example, Valeriana, or there are.
It might be a plant. It might be a plant that we don't hear.
I'm trying to think about plants that I know the names in English, like ginkgo. That
Oh.
you know that people take it and it's affecting a specific disease. But which ginkgo you're giving? Does it matter if it's grown in China, in Russia, or in California? There is one type of ginkgo. Every ginkgo that we'll take will be the same for the same disease. It's the same idea until if we won't start to understand, stabilize the genetic of the plants, understand what they are containing, understand how they're active, which strains or which one are effective, we'll stay in something very flat in herbal medicine, but we can take it to a next level, and that's what we're trying to do.
So are you working with strains? Do you break out the cannabinoids in the strains to combine them for precision medicine? Or are you, I think I read that you're documenting something like 900 strains that are grown in Israel, to be able to understand all
That's
the
true.
compounds. So is that how you're looking at it? Is one specific strain, or do you break it out into?
So usually I'm starting with the plant and breaking it down in order to prove that what I suspect is working. But a lot of time it's complex. So it's very difficult to break the plants and as a medicine, because if you need 5 compounds, it will be very expensive and very complex. But I'm breaking it down in order to learn which compounds
Yes.
and how it works. But then if you're asking about the lab, I'm breaking it down in order to understand. But if I think about the treatment, in a lot of examples, I think it must stay as a plant because it's too complicated to go with that to the FDA like that and things like that.
Oh, right. That was going to be my next question is, right, how would that work in the path to a pharmaceutical, I guess, let's say. I would imagine it's different in Europe, Israel, and the United States, but like I've heard that pharmaceutical companies might not be interested because there's so many different compounds that they just want to be able to take one compound. How would that work as far as the future of precision medicine, somebody coming out with like an Epidiolex or something?
Yeah, pharmaceutical companies want to make money. If you will convince them that 5 molecules will make them money, they will go with 5 molecules. They are not sensitive to they don't have opinion in there, okay?
Okay.
And I must say that, you know, there is a feeling that pharmaceutical companies want to block the cannabis research, but this is not true. They're sitting on the bench until they think it will be worth to get in and to take it. The regulation, the FDA regulation, we said different in Israel, it's not different. In the end, we all lay on an FDA approved drugs.
Okay.
So the regulation, the FDA don't like this complexity. So one molecules, it's easier to understand toxicity, the interaction. Then when you're going to 2, 3, 4, it's become more difficult and more complex. But I believe that in the end, in the other side, doesn't matter if it's FDA, whatever, there are people and humans that sitting, and it's education. They're learning also that this complexity have advantage, and they're learning it maybe takes more time. And we will start with 2 molecules, 3 molecules, and we go to 4 molecules, 5 molecules. If you ask me to the future, I'm sure it will be open more and more. And the pharma companies, they going with things that they don't want, how much money you need to invest in order to cross the line and how much money you will get. It's still not for them, it's not, no, they don't think it's profitable yet to go the direction, but I think it's changing. When I'm coming with the science that the physician can understand and the pharma company can understand, it's become interesting to them. And I have already offers for the things that I developed from pharma, because for them, if they can make it a medicine and sell it for expensive, they will do that. But I think, and we're learning that this complexity, our body work in this complexity, we want to treat diseases like Alzheimer, cancers, and neuro diseases that we don't have solution in the last 100 years, it probably will be in a more complex treatment. And plants bring that within, you know, when people go to a shaman or to, you know, physician 1,000 years ago, most of the drugs were plants. And he didn't give one molecule. He take the leaf from here, a flower from here, a root from here, combine them together and gave it as a medicine. He create what we call polypharmacology, create complex medicine. And what I'm trying to see is to learn this and to understand the complexity. Cannabis opened a big window for that, to understand that and to prove that. Because the endocannabinoid is complex and it's in many different cells, we find out that sometimes few molecules affecting one cell, few molecules affecting different systems, few molecules affecting third system, and you're getting a full treatment when you won't get a few that. You know, we spoke about IBD, inflammatory bowel disease, like Crohn's colitis. It's a disease of our intestine that become a chronic inflammation. But the meaning of that you have cells from the intestine or other plants, other places, that being affected, and they release specific molecules that call the immune system to attack them. And we see that cannabis is very effective to these diseases. But now we started to learn which cannabis, and we find out that there are molecules that affecting the colon cells or the intestine cells not to release the molecules, but there are other molecules that affecting the immune cells not to attack them. And if you combine them together, or if you have a plant that combine them together, you get a very effective treatment, because for one hand, you're affecting the colon cells not to release the cell. From the other hand, you're affecting your immune system, and you're getting a full or very holistic treatment and not just very specific. And I think we will go with that, and we will understand that, and we will find more complex medicines, whether it will be a medicine or still a plant. We just need to understand and to define which plant, which combination, which plants harboring the right combination to treat now colitis.
So amazing to think of it on that level. Maybe that's the missing key to these treatments
So
for
I have a talk that sometimes I'm giving conferences, and I call it, what is the 3 levels of the entourage effect. So when you're looking on one compound that binding one receptor affecting, this is the medicines today. One level of the entourage effect is when few molecules binding one cells and affecting in different direction on one cell. And we have examples like that from cancer and other, in specific cancer, in leukemia, we find out that in order to kill the leukemia, we need 3 cannabinoids that binding 2 different receptors, and we learn why they need 2 receptors. And this is first level of the entourage effect. This is actually what Rafi Meshulam showed for the endocannabinoids in 98 and called it the entourage effect. He showed that in order to activate CB1 receptors, you need 2 endocannabinoids to come together. Okay, this is the classic entourage effect paper by Rafi Meshulam. This is the first level, classic. Second level is what I explained to you in 3 minutes ago. Second level of complexity, few molecules, or one molecule is affecting one type of cell, other molecules affecting different type of cell, but they are working together in creating the disease. So you're affecting the disease from 2 different systems. One is the immune system, one is the colon system, and you're getting better effect. This is the second level of the entourage effect, which we usually not treating diseases like that. We're starting in healthcare to start to think like that. Yeah. And the third level of the entourage effect is more related to, I would say, quality of life of the patient. Because if you have a patient which have PTSD, let's say, okay, post-trauma, you have a problem to sleep, you have anxiety, you have bad dreams, we have few things that affecting. And you can have one plant that affecting the sleep, affecting anxiety, affecting other things. So you are treating holistically the patient, okay? I will give, again, sorry that I'm going to cancer all the time because this is my major work.
Yes, no, it's, I'm so,
yes. A cancer patient that now getting chemotherapy, he will suffer from anxiety, from bad mood, he will vomiting, nausea, pains, problem to sleep, all of that being getting it. But cannabis affecting all of that. So few molecules will affect the sleep, and we already learned which molecules are now. Few molecules will reduce pain. Few molecules will affect anxiety. Few molecules affect appetite. And now you're getting from one treatment, one plant, something that the patient need to take 5 different medicines, but you have it in one plant, and you improve the quality of life of the patient. This is the third level of the entourage effect.
Now, do you find that that's so interesting, the different levels? You just don't hear people talk about that. It's amazing. Do you find that THC versus terpenes, like is that a big part of the combination that we're talking about in the cannabinoids? Is a terpene a cannabinoid? It is a
No, terpene is a terpene. It's a
terpene
different
is a
molecule.
terpene. Okay.
The structure is a little bit similar. They're coming from this, sometimes from the same molecule that being produced, like, their father is the same father, I will call it like that, but
Okay.
it's a different molecule. Terpenes are existed in all plants, let's say limonene that are existed in cannabis. It's also in lemons or also in lemongrass, okay, or other plants. It's the same terpene.
And can
And
you verify, there's a lot of controversy in the United States about are the terpenes the same in botanicals and any other plant versus the terpenes in a cannabis plant? Is there, because a lot of people reintroduce terpenes from
no,
botanicals.
it's the same molecule, the same terpenes, the same effects, okay? There are other molecules like flavonoids or other molecules in cannabis. There are flavonoids that are unique to cannabis, like cannaflavin B, cannaflavin A, but in terpenes, we didn't discover any terpenes. We can see more than 150 different terpenes in cannabis, but we didn't discover any terpenes which was unknown from other plants. All the terpenes was known and the same structure. And terpenes is a big question, because the question, let's say when you smoke cannabis, how much terpenes really getting to your body, because they will burn, they are very, volatile, and how many, and we did few experiments when we took blood samples after people smoke a lot. I don't know if it's for quoting here, but I have a student that smoked in a row 5 joints, and then we took a blood samples today, and we see traces of terpenes, okay, traces after 5 joints. So the question is how much, if you take the terpenes isolated, okay, let's say myrcene or whatever, and you will test it, it's have an effect, it's an active molecule. But how much is really left when you're smoking cannabis? How much is really left after you're doing extraction? And how much it's part of the entourage effect? It's a big question. I can say that for sure terpenes affecting sleep, okay? Yesterday we're doing a clinical trials on something, and yesterday I went to the farm that's growing the specific strain that I need. And the people that pick that said to me, Daddy, you must test it for sleep and for appetite, because just for walking with that, we feel tired and it's increased appetite. So what they are smelling is just the terpenes, not the cannabinoids. So the terpenes are active, they're no doubt. And Ethan Russo is very big on that and have a lot of amazing work and studies on that. My question is, it part of the effect that we see when you're smoking or using cannabis, because you're getting traces of these terpenes to your body. But they are sure effective. They can activate the endocannabinoids system too. We know that. It's a question that I don't have good answers for that.
So we know that it can activate the endocannabinoid system, but we just don't know how.
We don't know how, we don't know. I don't have an examples from my study that we saw an effect, and in the end we succeed to isolate, and it wasn't cannabinoids, it was just terpene or a combination with terpenes. So I didn't prove that yet, even though, as I'm saying, if I would take specific terpenes and add them in higher amounts, more concentrated, they will be effective on sleep or other pain or other things. But when I'm taking a cannabis strain and I extract it or see what the amount of the terpenes there, I'm not sure how much they're playing a role. So I don't have a good answer. Again, I trust other scientists that I really appreciate, and I named Ethan Russo here, that did the very good work on terpenes and showed and proved they are effective and important.
And so all the different plants, the different strains that you use to test and treat and trial, that you're really relying on the THC to treat these different, like, cancer
No,
specific?
it's almost never the THC.
It's almost never. So what
Never.
is it then in the
First of all, I'm trying to run away from the THC. I think it's a problematic molecules to treat because the psychoactive, you have a limit of dose that you can give a patient. If you want to keep the quality of life of the patient, high THC, it's a problem, okay? All the Rick Simpson or the things that giving, it's really hurt the quality of life of the patient, He will be totally stoned. There is the THC can cause hyperemesis and other, things. So when I can avoid THC, I'm trying to avoid because then it's easier treatment on the patient. It's less effective psychoactive. In almost all the studies that I did, whether this is inflammation, different types of cancers, migraine like headaches, endometriosis, things that we did, it was never the THC as a major active compound. It was CBD, sometimes it's CBDV, THCV, but also minor cannabinoids that you don't have a name for them, A minor cannabinoid called 37315, I don't have a name for it, is blocking the estrogen, and this is the reason it's affecting this and that. We have other minor cannabinoids that reduce headaches, migraines, but it wasn't the THC usually.
Okay, I from what I've ever read, I've read that in a medical perspective that a low THC dose is very important to this combination. So it's very, to treating something. So that
I think in pain indication, the THC is playing a role. It was shown in autistic kids that reducing anxiety, it's important that very low amount of THC will be existed, like 0.5% or 1%. You have 20% total cannabinoids, but from that 0.5% is THC. But it was shown that it's still needed. I don't have the explanation why. It's part of the entourage effect or things that they were working together. But in many indications, you don't need it. You don't need the THC at all. It's other cannabinoids.
So the minor cannabinoids in the plant, which are mostly all the other ones except for these big ones we talk about, like CBD, do you think manufacturing them synthetically is okay? Or would you, like, if you had a preference to make medicine, based on a specific strain or cannabinoid, would you prefer to get it completely from extracting it from the plant? Is that
so? So I'm not against synthetic in general. I think a molecule, it's a molecule. So if you know how to make it, it's fine. The question, do you know really? Do you understand really the structure? And sometimes we said minor cannabinoids, but if I have a strain and I have a strain with 17% CBG, so it's not a minor anymore, right? If you have a plant with 12% THCV, it's not a minor, it's become the major. And genetically, people succeed to do breeding and to do things to create strains that have very high CBC, CBDV, THCV, CBG. There are already few cannabinoids that we call them minor, but we have strains of cannabis which they are highly expressed in them. And so if you have a molecule that is synthetic that can help somebody and improve his life, I'm fine with it. I don't think that our abilities today to understand the structure of these molecules and to make them synthetic is good enough to replace them with synthetic molecules yet. Doesn't mean that in the future, if it'll be cheaper and will be more safe and help more people, I don't. It's not emotional for me.
Yeah.
It's not coming from that. I'm not in love with the plant or the molecule today. I just
the outcome
want to help people. And again, I think cheap medicine, there is a meaning for that, I think it's important. I think this is totally different conversation when we are talking about animals. We are not investing money and time as we're investing in humans. So dogs and cats with cancer, usually you won't give them immunotherapy that costs $10,000, or even chemotherapy that cause them to vomit and to lose their hairs and whatever. You don't treat them. But if you will have a cheap medicine that's working, then there are another side of that for animals, another which is also, something that I guess it's important. So somebody asked me, but why do you think it would work in animals? I said to him, are you crazy? All my studies on mice and rats. Like, of course it works on animals. The question, does it work on humans? Not everyone works on animals.
And are you able to do any, I mean, we're not in the United States. Are you able to do research on clinical trials on humans? Is that, or is this all mice related trials?
So the question, how you define the trial, okay? So first of all, most of my work, I have a laboratory at the Technion, which is Technology Institute in Israel, and most of my work is on cells that are growing in a plate. We take cells from humans, we grow them in a plants, we create organoids, like mimicking, let's say, the colon, things like that, or we're working with mice or rats. This is most of the work. But when you discover something, now you need to test it on humans, and then you're going to clinical trials. There is classic clinical trials, which is double-blind placebo, FDA approved, and everything. And of course, we're going also in this direction. But especially in cannabis, patient already getting the medicine, right? I just need to tell them what to take, and then I can follow up on this patient. And this is something that we're doing. So if I define a specific strain that I think affecting sleep more than others because these compounds, I can go today in Israel, okay, and say to the patients, through the pharmacy and the healthcare, and get approved from the healthcare, or everything being approved, and do a double-blind placebo or a placebo trial, that patient getting the strain that I want them to get, whether others, and then to compare it. So it's a trials on humans, it's clinical trials, but it's not a classic. I'm doing kind of follow-up on patient. The patient get a prescription, at least in Israel, but I think all over the world, it's not written in the prescription, take these strains with these compounds. It's written cannabis. And then the patient go to the pharmacy. In Israel, in the pharmacy, we have more than 950 different products. He can choose whatever he wants, right? He didn't know what he wants. But if the professor is coming and said, hey, for your cancer, I think you should take that. Now, when he's taking that, I can follow up on I'm not forcing him to take, I'm not there, I'm suggesting it. But now I can follow up on patient that taking this product, whether this product, and test all the parameters that I want. And this is something that we're doing a lot. We did more than 25 different trials like that, following up on patient that taking cannabis, specific cannabis, that we know exactly the compounds, exactly then follow up on the patient. And it's blood samples from this patient, it's all the parameters. So this is something that shorten the times in years and money. Instead of investing $50 to $80 million in 7 years, you invest in $100,000 in 1 year, and you have good results on humans, which is a good starting point. And I think if we spoke of pharma companies, I think when pharma companies see that it's already work on humans, whether it's the whole plant with that, this is a big jump, because now it's working, it's working on humans, you understand from me what are the compounds, and what I think the mechanism of action for the pharma company is the big jump. So I can give a sleep as an example, that a pharma company that worked with me said, after you will show that it's working on humans, we will step in. And I just finished, we're writing now the paper on 120 patients that get this specific strain, and we test all the parameters on sleep and everything with placebo, and we saw that it's working. But then it's starting to be interesting for pharma.
Mm. And also, you're probably creating a database on all this for the pharmacists to use to be able to recommend to people specific
I'm
type.
trying. It's a whole mess out
Yeah.
there. When you have 950 different products, which changing, you import from here, you import from there, you're growing a new strain, then it's very difficult to follow up. When I started working cannabis in 2014, I got a big funding for a foundation in the state. I got $5 million to do follow-up on all the patients in Israel. And the reason was because there was just 80 products, 80, just 8 growers that was authorized to grow them. And there was like 15,000 patients. So it's something that was doable. It was 80 products on 50,000. If you have enough money. So we had agreement with the Minister of Health and the growers that every time that the grower pick a strain, make an extract, every time that there is a product, it's coming to my lab to analyze full analysis for all the cannabinoids and terpenoids, and then we follow up on the patient. So we have a barcode for every batch, every time that the strain that the grower pick a new batch, we had a barcode and we knew exactly what the patient is taking, and we combine this data together. But when it started to grow, today there is 150,000 patients in Israel. With 900 products, it's very difficult for me to do. It's too much.
But it's a beginning, to prove to people that we're starting to document this. And then I know we should wind this down because it's a long time, but I just kind of wanted to touch base again on the conversation that we had in Atlantic City is, so we're pretty far away, I think you were saying, from being able to look at testing someone's specific endocannabinoid system to identify any deficiencies and then say, oh, you should have this strain to treat your treatment. Is like if you took a blood sample of my blood sample, would you be able to find deficiencies? Are you working towards that, or you're just working on specific medicine like cancer, breast cancer, or where are we with that?
I will separate the two, okay? First of all, I don't believe that from DNA sample, you will be able to define which cannabis you need to take. Because first of all, it doesn't make sense to me that you will take a baby 1 year old today, take his DNA, and know in 40 years when he will have a PTSD what he needs to be treated. Because the DNA won't change, right? The DNA will stay. So if you will know in 40 years, you should know that. So the DNA is not changing, but we are changing in many parameters, and the PTSD that you're getting is not from your DNA, whatever. So DNA, it's, as I said, it's a cooking book how to make proteins, but it doesn't tell all the story, and it's way more complicated. Second, endocannabinoids are not proteins, so the DNA is not coded to endocannabinoids just for the receptors. If you're taking blood samples, let's say from your arm, you see the endocannabinoids that in your vein now in your arm, but the endocannabinoids in your brain is different, and in your heart is different, and in your skin is different. And if you want to take a real biopsy from the tissue that you want to treat, it's more complicated. But even after that, even I will take, let's say, a sample from your skin, which you have now a disease on the screen, which been created by the endocannabinoids, I still don't know the other side. I don't know which cannabinoids will mimic that, or which cannabinoids is for treating that. I'm doing a lot of work. I invest a lot of time and efforts to get blood samples from diseases that I thought maybe there is a correlation for deficiency of endocannabinoids. Autistic kids, epileptic kids, Alzheimer, and many diseases to try to define. I find it very, very flexible between different people. The bars are so big between people, which I find it very hard, very different between men and females. But I will Given another example why it's very complicated. There is a show in the Israeli TV called Trust Me, I'm a Doctor. Okay. This show, they are going with 5 physicians and they are testing things that people believe if they are true or not. Okay.
Oh,
If
okay.
you're going outside with, not dressed enough and it's very cold outside, whether you're catching a cold or flu or whatever, that is real. Okay.
Right.
There is few things that we believe, but the question is there is something. So one of the show was on the endocannabinoid system, and they came to my lab and to my company, and we did few things, and we showed that when you're running, running 3K and coming back, your endocannabinoid is changing because it's control your heat balance, it's control your oxygen, but also when you're feeling good with yourself, it's part of the endocannabinoid system and everything. So we showed the endocannabinoid before the run and after the run, 10 minutes run, how the endocannabinoid system is changing, which families, and we said these families probably controlling the heat balance, this family, then this is for fun. And then we took another group of people, different, and let them just to sing a famous song in Israel, and they were excited, and the TV, and then we take the blood sample before they sang and after, and we show that now just this group of endocannabinoids change the group that when you're feeling good with yourself, because it's not that they're not balancing the heat here and not balancing, it's just a group of them.
Yeah.
But this is teaching me how difficult it is to follow the patient, because if he coming to take a blood samples, even if he just sing in his car, or he got a good, his wife called him and was very nice to him, and then I tell him I'm waiting for an out, it can change his
mood. Yeah.
endocannabinoid. So it's so difficult to follow up on that. There is diseases called endocannabinoid deficiency. And again, Ethan Russo was there. But this is a very, very, very specific diseases on a specific mutation, on a specific receptor. It's not for the whole population. I won't be able to take a blood sample from you and to show now that, your emotional things changing. Let's say we are extremely far from that point now. Maybe we will develop more accurate measurement. We will learn more on the endocannabinoid system and also.
Well, I guess to wrap it up, I would just want to ask these 2 questions is, how close do you think you are in your work with cancer? I'm not saying cure cancer, but how close are you to getting to something, very that could really help? And the last question would be, what's your favorite cannabinoid right now, or most promising?
So I will start from the first question. Cancer, it's a given name to more than 1,000 different diseases. Glioblastoma, which is our brain cancer, breast cancer, and pancreatic cancer are different creatures. If I will compare it to animals, it's an ant, it's an elephant, and a bird. Okay, you can kill them with the same tools. Okay, or you can treat them with the same tools. It's a totally different diseases. In each cell in our body, there is its own types of cancer, and each one of them, there is different types. As we said on breast cancer, there are estrogen plus, triple negative, hair plus, and BRCA mutation. There are many types. So I think we in my lab succeed in 3 times from these thousands to find specific types of cannabis that can affect this cancer or treat the cancer, kill the cells, and things like that. Not on palliative care, really replace chemotherapy and things like that. But these are 3. This is very personal. I think in the immunotherapy, which I told before, I'm starting to produce products in order patient to take it, because this is even not a commercial thing. I feel a duty. I know that there are patients that getting cannabis and killing themselves instead of taking cannabis and curing themselves. and it's just tell them take this and that. So I'm putting a lot of effort that, it will be on the shelves, at least in Israel. It's a very difficult on regulation what I can say and not say, but I can't say this will cure and not cure until I did an FDA approved. But on the other side, I can stand from the side, see patient taking things that really harm them. I'm not talking about now you are suffering from pain and taking the wrong medication. You are dying and taking the wrong medication, friends of friends and family, and then I'm getting 2-3 calls a day. And because the product is not on the shelf, this specific strain, I can't find it. So this is something that I'm doing, I think in 1 month, it will be already out. So we're growing them and we're creating that and that.
Wow.
How far it will I reach to send it abroad and things like that, it's a big bet. And there is again a legal question here, how to do that without, to be sued by people or by them, but I'm doing that. On the other 2 cancers, which are breast cancer and leukemia, we are on the process to create that. I hope that in 1 year to 2 years, it will be available for the patient, but from the study, the research, we almost done. Now it's to take it further, how to reach the patient with it. It's not trivial at all.
So, okay, so I guess our last question, and this has been amazing, is what is your favorite, promising cannabinoid?
So there are few rare cannabinoids or minor cannabinoids that I find them extremely active and in different diseases, and I believe they will become major in the future. They don't have names because people don't know them. It's 3315 and things like that. In the known cannabinoids, I see that CBG is a very potent cannabinoid in many effects, wound healing, in types of cancers, in the immune system is very active, and I think it will be an important cannabinoid. First of all, it's not psychoactive. It's highly expressed in specific strains. There are already strains with 20% CBG. Wow. It's very, very safe. It's already been done safety on CBG, FDA phase 1 safety on CBG. And so we know it's extremely safe, not psychoactive, very potent, very effective on many diseases. And I think it's a very important cannabinoids. I'm not sure if it's my favorite, but I really believe CBD will become more and more important.
Oh, wow. Thank you so much. Wow. Thank you so much. This was so amazing.